Good day, everyone. On behalf of BioIT World's Global Web Symposia Series and our sponsor, Agena Bioscience, I'd like to welcome you to Accurate Pharmacogenetic Characterization of Challenging Samples, Management of Low-Quality DNA, and CYP2D6 Hybrid Alleles. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now I'd like to introduce our presenter for today. He is Robin Everts, PhD, Staff Scientist, Scientific Affairs with Agena Bioscience. Welcome, Dr. Everts. The presenter ball is yours.
Welcome, everyone, to this webinar, wherever you are located in this world. So my name is Robin Everts. I'm a Staff Scientist at Agena Biosciences. And over these past years, I have developed over thousands of assays in different applications. And the most recent years, I've actually worked a lot on pharmacogenetics. I've developed over 50 different panels for pharmacogenetics here. I also have been part and taken part in the GetRM project for pharmacogenetics. And during this time, I have been in many labs where I talk to people, and some things have come up regularly in the different labs. And the two of those that we're going to talk about today are actually low-quality DNA and the hybrid alleles. But before we dive into that, let's first look at why pharmacogenetics is very important to us. So we all are aware of adverse drug reactions. When we normally talk about the leading causes of death in the United States, we talk about things like heart disease or cancer. It might surprise you that adverse drug reactions are actually the fourth leading cause of death in the United States if you tally them all up. There are about 4 billion prescriptions a year, of which more than 2 million result in an adverse drug reaction, and of those, there are 100,000 people who actually die each year because of the wrong medicine. If you even divvy this number up even further, that means every 5 minutes somebody is passing away because of a bad first drug reaction. And also, anybody who has an adverse drug reaction actually has a much higher healthcare cost. So a lot of people have really been interested in getting into the pharmacogenetic testing because it can hold a lot of promise in helping patients. It is estimated that at least 75% of Americans have actually a genetic variation that can affect response rates to medication. And of course, the mission of us is to really help with pharmacogenetic testing to help you and physicians to customize therapies, so getting you the right drug, the right dose at the right time, which actually produces a much better to optimal outcome, and it can help minimize the adverse drug reactions. As you can see on the screen on the right, a lot of people have started seeing this, and it is actually predicted and shown up till now that between 2050 and 2020, there is about a 25% growth in PGX testing volume requests. And when the population is growing, this number grows, and also a lot of labs actually, they start looking into pharmacogenetic testing and getting a higher run rate. So they face rising sample numbers, and still some of the things they have to really take care of is turnaround time. How fast can I get my results back to the patient? What is the best way of reporting this data? And also, how can I do this at a reasonable cost? Primary consideration actually has to be also given to accuracy, because you can do all these things like turnaround time, reporting, and cost control, but if you don't have accurate data, the report means nothing. So it's really important to look at the quality of the data you provide to the patients, and there are several unique challenges that PGX labs have that can affect the accuracy if they're not addressed properly. And two of the things we are going to talk about today are DNA quality and the CYP2D6 hybrid alleles, which are lesser known. So first, let's look at DNA quality. So the DNA does not magically appear at the benchtop for an analysis. There is a whole stage of, or a whole project ahead of it, and there is kind of a chain of events happening. First of all, the sample comes from the sample collection site, and that can be at home or it can be at a hospital. It can be a different sample type. We're talking about buccal swabs versus, say, sputum or saliva. as I said, collection location, somebody can do it at home or it can be done at a hospital. And then also adherence to protocol. If the protocol that you supply with your kit says dry the swab for 2 minutes, does the person actually dry it 2 minutes? And then storage, like how does it get sent over to the clinical site? Does it get in a very hot truck? Or, for instance, like everybody knows the story about the kid who ate an Oreo cookie, and then you get the swab in with a little note on it saying, sorry, my kid ate an Oreo. Hopefully, you can still get some good results out of it. So that's the part where the lab has very little control over. Then the second part is the DNA extraction and purification step. There are different extraction methods and elution methods, and so depending on which extraction method or elution you use, it can also affect the outcome. And lastly, something in the lab while you run the panel, how is the assay designed? How robust is your panel to lower quality DNA? So there are a lot of steps that are affecting the quality, and it's really important because, as you can see on this slide, on the y-axis, we're talking about the number of NA calls or no genotypes. And on the x-axis, you see different sample types. The middle part are HapMap samples, and the bookends are actually clinical samples. And what you can see is you can design a panel on HapMap samples works really well. But we all know that clinical samples have a lower quality. So we have to be very careful in designing these because in the end, it is not HapMap samples you run, but it's clinical samples that you are running for patients. So because of the poor quality, you see a lot more dropout, and you can see here samples having dropouts up to 60%. What does it mean? If you have 60% dropout in your sample, it actually means that you either have to do a plethora of things. You can either try to get a new sample, try to run it again, try to do something different, but all these things lead to an increased turnaround time and also to an increased cost. So this is very important because with the rising number of samples, it is very important to make sure that we don't have a lot of these glitches happening. So this is something, while I was in the laboratories, I saw a lot happening, and I was very interested in learning more about this. So we here at Agena, we actually started looking at collection device handling as a first experiment. So we took several buccal swabs, we swabbed a number of people here, and we extracted those using a standard DNA extraction kit. What we did at first was we eluted these DNAs from different people with water, low TE buffer, or regular TE buffer. And we used it, or we analyzed it using standard panel here. We also did look at the same type of swabs and same people, but we actually stored these swabs improperly. So we stored them wet, we did not dry them down as according to the protocol for 6 or 13 days. And we compared those also against FFPE samples, which are known low quality samples. We did the same extraction kit, we eluted it with low TE buffer only and analyzed it on the same panel. So what we can see here is on this page is actually the outcome of the samples. So in the first box, we see the samples that are extracted using water, low TE buffer, or TE buffer. And you can see if you do the storage properly and you do the protocol properly, all these samples give very high call rates and extension rates. So these are all working really well, and the type of elution buffer did not matter. If you then looked at the samples that were stored improperly for 6 days or improperly for 13 days, you can see already start to see some discrepancies. So even at 6 days, the overall looks pretty okay. You can see a lot more dropout in the bottom, but after 13 days storage in the wrong conditions, you can clearly see that these samples are really, really poor quality. They are so poor, if you compare them to the FFPE samples, which are known poor quality sample, you can see that even storage after 13 days is way worse than a FFPE sample. So having seen this and seeing that if you follow protocols properly, it should work fine, we then went on and we took actually a third protocol where we took the polyester swabs from 17 volunteers. We selected 5 different commercial kits that are available everywhere. And what we did was each person gave 10 swabs, and we randomly assigned 2 of those 10 swabs to each kit. DNA was extracted according to the protocol, and we tested parameters such as call rate and extension rate. So the call rate really tells us about the sample, how many of the SNPs did provide the genotype, and the extension rate really shows more of how good this assay was performing based on this extraction technology. And this can help us identify sample versus assay issues. So when we analyzed these different extraction technologies, what was really easy to see on this slide on the left side was that kit A and kit E had a lot more problems than kit B, C, and D. Kit B, C, and D had very good extension and very good call rate. Kit A and E really had a lot of dropouts. And we thought it had to do with long range, the longer fragments. So we specifically looked at CYP2D6 because CYP2D6 has some longer fragments due to the pseudogenes that are available or present, and we want to make sure that these products were specific. So if you look at the graph on the right, you can see that the majority of these lower yielding dots were actually the CYP2D6 assays. So what we also then did was we were looking at if there was a correlation between the extraction method and success rate and the length of the products. As you can see here on the left, for method A, which is a lot short fragments, the call rate was 100%, was very similar to the method B. However, you could clearly see that after about 400 to 500 bases fragments, the genotyping success rate dropped dramatically. And after 800 bases, they were completely gone. There was no genotypes observed in longer fragments. Whereas if you look at kit B, from 100 to 1200 bases, these fragments all extended really great. So we could clearly see that some of these kits actually seem to be fractionating your DNA. The other thing that was also very apparent was we had 5 different kits, A, B, C, D, and E. And as you can see, they all had very similar OD readings. So based on the OD reading, we had expected these samples to act, or these kits actually to perform very much similar to each other. However, if you look at the call rates in the third column, you can see clearly differences. Kit 1 or kit A was really the poorest performer. Kit E was second, and the rest was like really good. So I'm not saying here that one method is bad and other ones are good. It really is about that solutions may actually differ between labs and DNA sources. So it's really something that you have to double check. And especially for PGX, I do believe PGX is more like a system instead of like one thing only. You have to look at the whole picture from DNA extraction up until the resulting reporting. So what we recommend normally to laboratories is if they are starting out with PGX, take 15 samples that you have used for your current method and make sure that they work with your new method also. So just have known samples there that you are aware of what your expected outcome is. If you have never done PGX before, you have no technology in-house at all, I mean, we normally recommend you try a few different kits and see which kits work best for you. So you can start out with the best available data there. And yeah, some of the things we normally see is really like the common issues are, if it's a specific assay that's affected, well, the first thing you can do is like check the Pcr product sizes. And if you see that longer fragments are mainly affected, the thing you most likely have is an extraction kit that really fragments your DNA, or there's a sample type that's more prone to degradation due to the storage or the how it's shipped to you. If the call rate is more randomly affected, then most likely you might be able to switch out your elution buffer and see if water is better than your elution buffer, because that sometimes also affects the downstream applications. So we talked about the DNA quality and really about the things we can do there. The other thing that can be done, of course, is also looking at the panel. So I would like to introduce here the Veritas core panel from Agena. This is a panel that targets 73 different targets in 20 genes. And it's a very robust design. This actually was developed using the samples, as I shown here before you, and many others. What we did with this panel is we really looked at what the amplicon length is going to be. We optimized the amplicon length to make sure they were as short as possible and still unique and specific. We looked at extension primers, making sure that they were the best ones we could find and giving the best results. We looked at multiplexing capabilities, checking which assays would work best together. And we also sometimes changed some component concentrations. For instance, that Pcr product that's a little longer, we might give that a little bit more Pcr primer in order to boost that efficiency there. And what you can see here is that we compared the same type of samples versus the previous data we have. And what you can see is several assays, like for instance this one out here and the first and second one, you see that the performance between the other PGX panel and the Veridose score is very similar. But for assays that had issues with longer fragments, you can see within the new Veridose score panel, they have very high quality calls. So we really have a panel here that is more resilient to very low quality DNA. And to show that more, here is actually the same table as before, where we see the original data, where you can see that 2 of the kits had a lot of dropouts. With the new Veridose core panel, most of these dropouts were rescued. One thing with the kit A, I mean, the problem there was these fragments were below 500 base pairs, and we had to have a few fragments a little over that just to make them still specific for CYP56. So with this new panel, we actually then went to some customers, and here's an example story of one of our customers. They were running PGX, and they had 62 samples that they have tried multiple times, and they were always failing. So they got these 62 samples run on our new Veridose core panel, and without doing anything, without any analysis, 48 of these samples got complete genotypes right away. So we actually reduced their dropout rate by 77%. So the panel Veridose core could rescue 77% of samples that were dropped out previously. And this would, of course, be a great thing for a PGX lab, because having less dropouts means less redo work and a better turnaround time and less cost. So this was the part that we talked about, the DNA challenges. Now we're going to go on to the second part, which is about the hybrid alleles. So let's first talk about CYP56 itself. So CYP56 is one of the genes in the genome that has copy number variation. There's about 4% of genes that have copy number variation, of which CYP56 is the most important one, because it's relevant for between 25 and 40% of drugs being metabolized by the body, depending on what literature you read. So within CYP56, you can have no copies whatsoever. So you are a 0 copy sample, or in this case, because the gene is not there, you are poor metabolized with certain drugs. You can have one copy or 2 copies. 2 copies is listed here. That is like the wild type. That is the normal state. And then you can also have duplication, such as 3 and 4 copies. And depending on which alleles you have, so it's not only copy number analysis that you do for CYP56, you also have to look at which alleles are present, which forms of the gene. And if you have multiple copies, there is a chance you become a rapid metabolizer, so drugs are being metabolized much faster by your body than normally. And from a recent study by Del Tredici et al. From Millennium Health here in San Diego, when they did a retrospective analysis on their about 100,000 samples, they found that CYP56 structural variations were observed in 13% of the patients, of individuals, and they accounted for 7% of the total number of variations observed, which is pretty high. This is another paper by Zhu et al. Out of 2017, and they actually looked at different ethnicities. And what you can clearly see from this picture and from this graph is that there is a fair number of difference between populations. For instance, like you can see in the first one, in Europeans, 51% of these have a STAR2 allele, whereas if you go to the third one, you talk about East Asians, and 68% of people have a STAR10 allele. So there's a lot of variation within each ethnicity. And also, if you look at the number of reduced activity alleles, there is variation there. If you look at the bottom here, Europeans have about 50% reduced activity alleles, East Asians 80%. Same thing for copy numbers. duplications and deletions, you can see they range from a low of about 4% in South Asians to a high of about 15% in Africans. So the reduced activity alleles are important, but also deletions and duplications clearly show a big difference between the different ethnicities. And outside of the duplication deletions, more recently, a lot of people have also seen hybrid alleles. So hybrid alleles are actually CYP26 alleles that at some point in their sequence have a unique rearrangement with other genetic material, mostly CYP27. And so you get a partial CYP26 gene and partial CYP27 gene. And because of this rearrangement, these are non-functional. And the estimates of these prevalences of these alleles varies. I highlighted here a 45% in Japanese samples. This is from a paper by Soyama et al. Out of 2006. They found when they did sequence analysis of Japanese patients that about 45% of them actually had a tandem duplication with a STAR36. A more recent paper by Chen et al. From 2018 that did study in Hong Kong, they actually found 53% of people in Hong Kong had a STAR10, STAR36 hybrid or tandem hybrid allele. So you can see in Asian samples they're pretty high, and there's also other hybrid alleles present. So let's look at the most common hybrid alleles that are present. As I said, STAR36 is the one that I just talked about. STAR36 and STAR4N actually share the common denominator, which is called an exon 9 conversion. So in these cases, the gray sequence is all CYP26, and the red sequence in exon 9 is actually a small exchange with CYP27. This actually renders that allele non-functional. As I said, STAR36 is very prevalent in Asian people. A STAR4N is more prevalent in your Caucasian people. The second hybrid allele that's prevalent is STAR68. It starts out as CYP26 also, and then in intron 2 it swaps into a CYP27 sequence. Because there's this rearrangement with CYP27, this allele is also non-functional. This STAR68 is mostly found with STAR4 samples, again in mostly Caucasians. And then you also have the STAR13, which in the past used to be a breakpoint between in exon 2 and exon 3, a kind of mirror image of STAR68. Nowadays, STAR13 denotes also other assays where it starts out as 2D7 and at some point in the sequence goes into 2D6. These are also non-functional, and we have seen these as a frequency of about 1 to 2%. So if you now go back to that picture from Zhu et al. About the East Asian people that I talked about, where they said 68% of people or East Asians have a STAR10 allele. Actually, 2/3 of those actually were found most likely to be a STAR10, STAR36 tandem hybrid. So they are more prevalent than we think they are. And how do people at this moment mostly go into looking at copy numbers? So there are two standard methods out here nowadays that people are using. One is long-range PCR, and the other one is competitive PCR. But both, in the way people use them, are normally not geared towards detecting hybrid alleles. Long-range PCR, most labs I have visited in the past, they actually only type for a duplication and a deletion, and they are predicated on the fact that the primers for PCR are CYP26 specific. So if you have a change with CYP27, there is a different sequence for one of the primers and it will not bind, so you will not be able to detect those hybrid alleles. With competitive PCR, what a lot of people seem to be doing is run only one TaqMan assay and get a call for that. And if you only look at one point in the gene, you might not actually know what's going on completely. And both these methods might actually result in over or underestimating gene copy number. And if you have the wrong gene copy number, that actually can result in an incorrect metabolite status. I want to give you two examples of that. On this slide is the first one. Suppose you're a lab and you run a exon 9 assay only. So if you run the the competitive PCR for exon 9, you might find only one copy of CYP56 present, the gray one, and you might call the sample star 10, star 5. Well, in actuality, this sample, the way it is actually star 10, star 36, but you were lucky because in both cases, these are intermediate metabolizers. If you had run only an intron 2 competitive PCR assay, you would have found 2 copies because there were 2 copies of CYP56 at that point, and you might have called the sample star 10, star 10, which is actually more like an extensive metabolizer, so there was an incorrect metabolizer status. So you could say, well, we should only run exon 9 assays because that seems to get the right answer. However, in this case, where you have a star 13 allele, if you run the intron 2 assay, you find only 2 copies there, and you would have said this sample is star 1, which has the same metabolizer status as star 1, star 13. If you had run the exon 9 assay in this case, you would have found 3 copies. And so you would have called the sample a star 1, X2, or an ultra-rapid metabolizer, whereas in fact it was only an extensive metabolizer, and this person might have gotten the wrong dose. So you can see neither assay is perfect, and neither assay run by itself really does give you the right data. This is not something that we came up with ourselves. There have been papers in the literature already, and this is a paper from John Logan Black at Mayo from 2012, and they did a retrospective analysis. They had a number of star 10 containing samples and a number of star 4 containing samples, and they actually took those samples and added more genotyping data, copy number analysis to those. What they found was that more than 20% of their star 10 samples were actually containing a star 36 hybrid. Also, for the star 4 containing samples, they found more than 20% of those samples actually had a star 68 hybrid in association with the star 4 allele. Moreover, when they looked at all the data, 1% of the samples that were thought to be new to N only, they actually had a hybrid allele. And 7% of the samples that previously already had shown to have a copy number variant, with an extra copy, they actually, in addition to that extra copy, also were carrying an undetected hybrid allele. And I've said hybrid alleles are mostly non-functional, but in this paper, out of these samples, they actually saw that what they had originally classified the sample as, for in this case, the first one was indicated as star 1, XN, so 3 or more copies. They actually, after they did the analysis with the other new assays, they had to reclassify the sample to a star 1 plus a 2D7 hybrid plus a star 1. So they actually had to change the predicted phenotype from an ultra metabolizer to an extensive metabolizer. And you can say, well, 25 samples, that's not too many. However, this is 25 out of about 1,200 samples. So between 1 and 2% of samples had to be reclassified. Say, to put it in perspective, if there are 100 people on this call today, and they all have genotyped 100 samples this week, then we would have detected 10,000 samples with a copy number. If 1 to 2% of these samples were misclassified, that would mean 100 to 200 people would get the incorrect classification and maybe the incorrect dose or the incorrect medicine. So it is, if you look it up, or if you sum it up, it's a pretty high number. So there are labs actually that try to address this challenge, and what they do is they genotype in one workflow, they run one copy number assay together with that, and then they will combine the data and see if there is a star 4 or star 10, and mostly a star 10 assay allele present, and only if there was a star 4 or star 10 allele present, they would actually run a different copy number assay in a different part of the gene in order to see if there was a star 4N or a star 36 present. So this is great. However, you already have 2 separate workflows here. Then you start adding a third workflow because you have to run for certain samples a other copy number assay. So this results in issues with turnaround timing. It results in issues potentially with cherry picking samples for this second assay, and it might actually reduce or increase the turnaround time that you have for your results. So we think that a more holistic view of the gene is required, and so that is why Agena came up with the Veridose CYP26 CNV assay. So we have a single well assay that we developed. It actually looks at the CYP26 gene in 7 different sites with 21 different extension primers, and it will actually give you the actual copy number for the assay, or for the gene, but it will also, with the software included, it will tell you whether there are hybrids and which of the 3 hybrids is present. And lastly, it will actually tell you what the number of functional copies is that we are having here. As I said, this is a one-well assay, and it can be run concurrently with our Veridose core panel on the same chip because it uses the exact same conditions. So how did we get to this panel? Well, when we designed it, we actually studied 1,120 Coriell HapMap samples, and many of these samples had data previously generated by TechMan, long-range PCR, and other methods, and we got it out of literature. We also internally did TechMan assays, and one of the things we found from the literature, many of the Asian samples that had data were just called star 10, star 10. They did not have any data on star 36s. So when we did the original analysis and compared our findings to what we knew, we had an initial concordance of greater than 90%. This seems low, but we detected a fair number of hybrid alleles in the majority of the discordant samples. So we had 146 samples that had a hybrid allele, and a fair number of these were unknown. So we actually believe that the Veridose showed more than 99% final accuracy. We also showed there were 13% of samples containing a hybrid, and approximately 70% of these hybrids were previously unknown. So if you put it in a graph here, what you can see here is that a little over half of those hybrids were star 36 containing hybrids, a little less than half were star 68s, and just under 1% of these were star 13s. And the majority of those were actually unknown before. And what we did, because of my work with the GetRM Pharmacogenetics group, I actually contacted Andrea Gaedick, and we sent her a few samples to test for us because she runs a lot of different technologies out there. And as you can see here, we have some samples which this was the haplotype we had known up front. This is what we thought they were going to be. And this is the data that Andrea Gaedick generated for us. And you can see that the data that we generated based on this panel was concordant with what an independent lab provided to us. So next up, we actually did another study where another lab provided us over 1,000 clinical samples, and we in-house actually ran the TechMan Intron 2, Intron 6, and Exon 9 assays next to our Agena Veridose CYP26 assay. So we compared the copy numbers and the hybrids where possible. So for instance, star 36 was easy to compare because they show a lower Exon 9 versus Intron 2 and Intron 6 on TechMan. So we could compare that. The star 13, we also could look into those because for star 13, the breakpoint is after Intron 2. So looking at Intron 2, Intron 6, and Exon 9 for the TechMan assay, we could deduce whether we had the same data. For the star 68, unfortunately, it was not possible to confirm whether the star 68 was present or not because the TechMan assay was after the breakpoint, so that we only had TechMan assays after the breakpoint. But if you look at the data we generated, out of those 1,050 samples, 1,022 were concordant. So we had an overall concordance of 97.4%. If you look at the individual groups, for the 1N samples, we had 100% concordance. For the 2N samples, we had 100% concordance. For the 3N plus samples, we only had a 90% concordance. But this is mainly because we included what we considered 3N, including the star 68 hybrid alleles, were in this group. So all the samples that were discordant were star 68 hybrid alleles. For the Exon 9 exchange, which is apparent in star 36 and star 4N, we actually were 100% concordant also. So we think that our our panel actually is much better than the 97.4%, but yeah, it's because these star 68 assays that are not genotyped normally is where the only discrepancy is. We also sent an aliquot out of this panel to some customers and had them tested out. So the first analysis done against competitive PCR. There were two sets of HapLab samples that were tested. One was 48 samples that were previously genotyped by us also, and we got a 100% concordance there. The second set was a set of samples from that lab itself, and when they ran the first analysis, they found a 96% concordance. 76 of 79 samples were exactly what they were expected to be. The three samples that were discordant, actually, we found a hybrid allele present that was not detected previously. And when we looked at the other data, we could clearly see that this was actually right, the hybrid allele. So the conclusion was that we have a greater than 99% final accuracy here, and we detected hybrid alleles that were not detected previously. We also compared the panel against long-range PCR. So we had a customer who normally uses only the DUP and the DEL assays. They also genotyped their samples with our Veridose panel. And what we saw, the initial concordance was 94%. 270 samples out of these 288 that they tested were concordant exactly between long-range PCR and our panel. 18 samples were discrepant. From these 18 samples, 13 actually were star 68, and 3 actually showed an exon 9 deletion. So the majority of samples that were found discrepant are actually hybrid alleles, contain a hybrid allele. So we have 9 samples that have been investigated and that we were proven to be right. The other 9 samples still under investigation. So taking that into account, we have at least a 97% final accuracy, but we expect our accuracy to be 100% if the last 9 samples also are confirmed. So again, we ran it against a different technology, and we picked up hybrid alleles that were not picked up before. So the conclusions of this presentation is that labs must be very cognizant of and take steps to address common challenges, such as low-quality DNA and CYP26 hybrid alleles. I hope I have shown here that the collection procedures, the storage, the DNA extraction methods, they all can affect the outcome of the data, getting accurate data, and you have to really be careful and make sure that you have a technology that gets you the highest quality DNA present. I've also shown you data here that shows that when you design panels, you have to make sure that they work on suboptimal inputs, because low-quality DNA is very likely unavoidable in pharmacogenetic testing. Besides that, I hope to have shown you also here that the CYP26 hybrid alleles are very important. CYP26 is one of the most genotyped genes for pharmacogenetics, and if you get the copy number wrong, that can definitely affect your outcome and your result in reporting. So I've shown here hybrid alleles are common across different populations, and you can see it varying from about 50% in Asians down to maybe 5 or 6% in other ethnicities, and there are also different hybrid alleles in different populations. So the common copy number detection methods right now that people are using, unless you run multiple different assays, you cannot detect the right copy number. So we really believe that broad gene testing for CYP26 and other copy number genes is really important with respect to these hybrid alleles. So I would like to conclude this seminar or webinar here and open it up for questions and answers. As you can see on this slide, I have put in my email address just in case you want to reach out to me personally. I also have put in the email address of my colleague here. And please send us an email if you have specific questions, and I would like to hand it back now to Elizabeth.
Thank you very much, Dr. Everts. Yes, we do have quite a few questions that have come in. Our first question, can you share the manufacturers of the 5 kits that you tested?
I don't have them in the top of my head, but please feel free to send us an email and request that. because I chatted into to the webinar, and we have written a white paper actually on this topic, and we can supply that to you. I don't have the exact details here.
Our next question, what analysis is required for the user to see hybrid alleles with the CNV panel?
So that's a great question, actually. I tipped on it briefly during the webinar, but due to time, I did not want to spend too much time. But the panel actually comes with a reporting software, and the software will do most all the work for you. It will actually look at all the individual sites, generate copy numbers for all the individual regions, and then does analysis whether there are hybrid alleles present or not, and it tells you, it reports out which hybrid alleles present, and also what the total copy number is, as well as the functional copy number outside of the hybrid alleles.
Our next question, what content is on your core panel?
So the core panel, as I stated, has 73 different mutations in 20 different genes. The details can be found on our website. I don't have them handy here. And again, if you are interested in that, definitely let it not be known on the chat function or email me and or my colleague Keith separately, and we can provide that information to you.
Our next question. The presentation showed a few instances where controls or other reference samples were found to be incorrectly characterized. Can you explain why you think that happens and if you believe it is common?
I think that refers to the slides from the Mayo study where they found 25 samples to be incorrectly characterized. I think up to now, a lot of people are only running a single competitive assay or just a long-range PCR, and that only gives you data on one point. It will never be able to tell you whether you have a hybrid present or not. And based on the data we have, there are about 13% of samples that we generate the data on, we had hybrid alleles present. So they are much more prevalent than people think. So there is a good chance that for a 2N sample that there are undetected hybrid alleles, because nobody is right now, or I can't say nobody, a lot of people are not looking for them. So if you don't look for something, you will never find it.
Exactly. Thank you. What assay design technique do you believe has the biggest impact for protecting against low-quality DNA?
I think the most important thing really is because low-quality DNA most times has to do with fragmentation. I really believe that limiting the amplicon size, the Pcr product size, is the one single most effective way. Of course, you have to be careful because you can go too small, and then, for instance, for CYP2D6, you might not be able to distinguish CYP2D7 or CYP2D8P1, which is also important. So it's a balance of small fragment size for the PCR and uniqueness of the product that you're generating.
Which, if any, of these hybrid alleles result in loss of function phenotype?
So that's a great question too. Every hybrid allele that has been found up to now actually results in loss of function. But again, it's because they're loss of function, it doesn't mean you don't need to genotype them, because it really is, they are part 2D6 and part 2D7. So depending where you do your copy number assay, you might actually pick up the 2D6 part also and get the wrong copy number there.
How many wells is the Veridose CNV panel?
Great question again. I mean, the Veridose CNV panel for CYP2D6 with the 21 assays in 7 different regions, it is a single well panel, and it can be run together with the Veridose core assay on the same plate, so there's only one workflow you have to worry about.
Excellent. You mentioned during your presentation that you believe that swabs that are stored wet have reduced performance. Why do you say that?
Well, we have the data in-house, and one thing that definitely happens, so what most times happens is if you look at these swabs, they have very nice canisters nowadays, plastic canisters, and if they are closed down, they cannot breathe, and the moisture stays in a capsule. So as long as if they're wet, enzymes that can still be active and can chew up the DNA, because that's a regular apoptosis reaction. So you really have to make sure that customers follow the right procedures and really dry these swabs So everything is kind of frozen down into place, and these enzymes cannot really keep on chewing away on the DNA. Excellent. How prevalent are hybrid alleles in populations that are not Japanese? That's a great question, and kind of it refers back to the other one. So we do know from like Asian samples that they can be about 30 to 50%, depending on who you talk to. Like the one from Soyama et al. Was, I think, 45%. I just recently had another paper that I read where it was 50%. But those are commonly tested nowadays, whereas with star 68 and star 13, the testing is much lower. In our case, we confirmed exactly what the group in Mayo found. We found about more than 20% of our star 4 alleles actually contained a star 68. So if star 4 is 20% of your population, you might have 4% star 68s also there. And if for the star 13s, we found about 1 to 2% of the whole set here had this. So, you know, it's different per population, per ethnicity, but there is definitely, they seem to be high enough to really be worried about them, because we normally genotype alleles in panels with sometimes lower than 1% frequency, and it seems like these copy numbers or the hybrids have sometimes a much higher frequency than 1%. Okay. Next question, can you explain the advantage of the Agena method versus the reflex PCR method you described? Yeah, sorry. So with the reflex technology, what you do is you run your samples with one assay first, one copy number assay, and then you look at the data from that assay together with your allele calling. And so you first have to go through the whole process. And then if you have a star 10 sample, for instance, then you pick that sample out of the plate and run it on a different assay. So there's two issues there that I can see happening right away. First of all, you have to have somebody who can pick the right sample from a 96 well or a 384 plate, depending on what you run. And then the next thing is that those samples then have to be run on a separate plate with a separate assay. So you add in another workflow for a subset of samples, and I don't know that can hold up your whole batch reporting or just for those samples. So you get like samples that get reported out first, and you have a bunch of samples that are not being reported out at that time because you're waiting for another data point. So you have to keep balance of all these different workflows, whereas with the Agena solution, everything is in one well. So you at that point have all the data at hand to make the call the first time. Right, and I understand that you do have the extraction information at hand now. Would you like to comment on that? I can definitely do that. So my colleague was so good to pull it up. So we had 5 extraction kits. So the swab we were using was the Puritan swab. And the kits we were testing out was the QIAm DNA mini kit, the Symo Quick DNA MicroPrep Plus, the Invitrogen Charge Switch gDNA buccal cell kit. Then we had the Stratic PSP Saliva Gene DNA kit. And we had a Measuring Nagel nuclear spin tissue kit. And for exact details on product numbers, etc., please shoot us an email. But we had a wide variety of different providers, and these were just picked because they were very used by our customers also. Excellent. We have time for just one more question. What are the gene locations that this panel looks at? So that's a great question. So as I showed in the picture, I did not really point them out that much. We have a few assays in the 5' UTR, and then we have exon 1, intron 2, intron 4, intron 6, intron 7, and exon 9. So we have 7 different positions in the gene and 21 assays overall that pinpoint the different regions and get a copy number for each of the regions. Excellent. Thank you so much. We do have a few more questions, but we don't have time for those, so I'll be forwarding those along to Robin and Keith, and you'll receive your answer via email. So with that, I would like to thank our presenter for today, Dr. Robin Everts. I'd like to thank the folks at Agena Bioscience for sponsoring today.
Most of all, I'd like to thank those of you who came and spent this time with us. We're very grateful that you chose to do that. We know you're very busy. Hopefully we were able to provide some answers that will make life in the lab a little bit easier for you. So on behalf of BioIT World's Global Web Symposia Series, I'd like to thank you all so much and wish you all a great day. Thank you.